Okay, so we're recording now. So thank you everybody for coming. Thank you, Daniel, for coming. So I'm Regina Alvarez, and welcome to the second of the Dominican College Science Seminars. I want to introduce Daniel Atha, who is a botanist over at the New York Botanical Garden. His title is actually Director of Conservation Outreach. Daniel and I met back in 2013. He reached out to me to help with a BioBlitz in Central Park. For those of you that may not know what a BioBlitz is, that's a 24-hour inventory of all organisms in sort of an area that, whatever area you pick. And so Daniel was tapped to lead the plant group, and that's a pretty big task in Central Park. So he reached out, and knowing, seeing that I had worked there, he asked if I would give him a hand. And I said, sure. And so of course we hit it off, and we were, teaching people about plants in Central Park, and decided we needed to re-inventory the park. An inventory had been done in 2007. We decided it was time to do it again. And so we got together and worked on documenting the wild flora of Central Park. The two of us and several other people, Ken Chaya, who's on this meeting today, and some of the staff from Central Park. So it was a really great project, and I got to know Daniel and learn a lot of botany from him. Let's see, he's done botany around the world, Belize and several other places. But more recently, he's been working on more local flora, which is exciting for me. That's sort of what's sort of near and dear to me. And he and the folks at the Botanical Garden started or developed something called the New York City Ecoflora Project. And so that's not just documenting the flora of New York City, but really engaging New Yorkers to participate in that process. And as I try and teach my students, when people participate and learn, it really gives you sort of a much more of a connection with the organisms that you're working with, and you enjoy the local flora more, and in that way it helps conservation efforts and preservation efforts. And that's what he's going to talk about today. So I'm going to let him talk about that. The New York City Ecoflora is going to have a conference coming up. What was the date? The date was
November 6th.
November 6th from 10
a.m. to 12.
I'm going to put the information in the chat, and I'm also going to email it to everybody. I have everybody's email from this, so I'll email that to you so that you all know about it. So if you can attend that, I highly recommend it. As Daniel's talking, if you have questions, please put them in the chat, and we'll do the questions at the end. And
we'll weed out all the hard ones, and
let's see who can stump Daniel. No, but seriously, if you have any questions, I'll read them out. And Daniel can answer them. So put them in as you think of them, or at the end if you think of them. And I think that is all I'm going to say, and I'm going to turn it over. I'm going to say thank you again to Daniel for coming, and turn it over to him.
Okay, well, thank you, Regina, for asking me to speak. I was so glad when you started your professorship there at Dominican College, and I was really looking forward to come and visit you on the campus. But I guess this is as close as we're going to get for a while now, hopefully soon. But yeah, going back to Central Park, I learned a long time ago to ask for help when I needed it, not to be too proud. And, Central Park's a big place, and I wasn't really that familiar with the plants there. And so I looked around for the expert on the plants of Central Park and learned that it was Regina Alvarez. So I was so glad that I reached out to her, and that experience helped to inform the EcoFlora project, because around that same time we were writing a grant for doing a flora of New York City to document all the plants in New York City. Somebody has to do it. Um, a botanical institution needs to, be on top of what plants are here, which ones are rare, which ones are invasive, new ones, so forth. It's a job that needs to be done, and that's a big job too. A lot bigger than Central Park, 302 square miles of New York City. And again, you know, I thought my colleague Brian Boom thought that we need to ask for, we cannot do this alone. The two of us, we could get 3 or 4 more botanists working with us. There are not enough botanists in New York City to be able to really monitor the plants of New York City adequately to keep up with so much change and so much happening here in New York City. As we all know, it's a city that never sleeps, and the plants aren't sleeping either. They're busy doing stuff, and we need all of your help to keep track of it all. We cannot do it alone. So we developed this EcoFlora project to bring all of you in on this enterprise. It's a community science project. We reached out to Regina with help of Central Park, and we need all of your help to keep track of the plants of New York City. So in my presentation, I'm going to go through how we're doing that and how you're doing that, how everybody is contributing together to keep track of the plants in our great city. So I'll start my presentation now. Oops, got to share the screen first. Look at my desktop with all that. Okay, how's that look, Regina? Nod your head. Look good? Okay, the New York Botanical Garden is an advocate for the world's plants and fungi that produce oxygen, clean the water, hold the soil, and make all other life possible. They give us bodily nourishment, and they provide inspiration and meaning to our cultural lives. As a collections-based institution with some of the most comprehensive botanical resources in the world, the New York Botanical Garden is uniquely capable of providing the community with science-based information on the plants and their ecological dynamics, informing management and policy so our planet and our city can be managed wisely and sustainably. The New York City EcoFlora combines the NYBG's historical strengths in collections with emerging opportunities in digital technology and community science to maximize the impact on plant conservation in New York City. All New Yorkers are invited to participate as community scientists to observe, collect, and compile information about the city's plants and their relationships with other organisms, such as birds, insects, mushrooms, etc. Superimposed over this wonderful map of the world from the National Geographic is a graph of the approximate number of species on Earth over the last 500 million years. So we all know the Earth is about 4.5 billion years old, right? So 500 million years is only, a small 10% of the history of life on Earth. But before those 500 million years, there were like 3 species on the planet, right? Some bacteria, and that's about it, a couple types of bacteria. Until about 500 million years ago, when life really started to take off, which you can see over on the left-hand side of that graph. So it's just a simple graph of the total number of species on the planet over 500 million years. And the gallery is over I hope you can see the very right-hand side of the graph. Um, Immediately obvious is the great fluctuation in the numbers, right? You see it goes up and then it goes down, it goes up and then it goes down. Each one of those great troughs are the 5 major extinctions of life on Earth. We've all heard about those, right? The 5 mass extinctions, the meteor that hit the Yucatan Peninsula, um, bacteria overrunning half of the planet, and volcanic eruptions and sun flares, all those other events that wiped out, 90% of the life on the planet. But what happens after each one of those mass extinctions? What do you see in the graph? It's constantly going upwards. It comes back greater than it was before. That's what I think is so beautiful and so amazing about life on the planet, is it can go through these mass extinction events and come back even more diverse than it was before. Part of the reason is because these extinctions have happened slowly over a long period of time, many of them, and they were selective in the organisms that they might have wiped out at any particular time. So there was opportunity for some resilience in certain groups and some opportunity for exchange, and dynamics in the support of these organisms could help each other get through this event and come back even more diverse, with the help, of course, of bacteria and fungi, which are transferring genes from everybody all throughout the biosphere. What let's see. The very tip on the right-hand side is after the last great extinction. That's the Holocene period, the last 60 million years since the dinosaurs were wiped out. You see that the diversity of life on Earth was off the chart, is 3 times what it ever was in the last 500 million years. And the very end of that graph, you see where it ends right there, that's where we're sitting. The Holocene, this last 10,000 years has been the most extraordinary flourishing of life that the planet has ever seen. Amazing diversity, which you can see on the screen there, scattered all over all the continents, just covered with incredible diversity of plants, animals, microorganisms, and so forth. And it's this diversity of life, the biosphere, that's created the atmosphere that we enjoy, or that we've been enjoying up till today. 78% nitrogen, 21% oxygen, and 0.04% carbon dioxide in the atmosphere. And we all know what's happening now, right? Where that graph is peaked all the way. Unfortunately, we know that that's a peak. It's not going to continue going up, is it? That graph of diversity of life on Earth, and the composition of the atmosphere is drastically changing, and it's all going in the wrong direction. It's everybody knows that. And we're now in the Anthropocene. It's a totally new geologic era, so dominated by humans that our presence will be detectable in the rock strata millions of years from now. There will be a thin layer of mercury and nickel and copper, and all the stuff that we're digging up out of the earth and putting up into the atmosphere is going to be in a thin little layer. All the rest of the stuff, the plastic and, all of our bones and everything will be all gone, but there will still be a signature of human presence millions of years from now. And that will define the beginning and the end of the Anthropocene, the era that we're currently living in. And what's really extraordinary is that we are probably the only organisms on the planet that have witnessed the transition from one geologic era, like the Cretaceous or the Eocene or the Cenozoic era, something, into the next geologic era. How many organisms have, it's never happened so quickly. It's a pretty extraordinary time to be alive. And it's also comes with great responsibility, right? It's not looking very good for future generations. So human beings set out to conquer nature, and in the process have unleashed the sixth mass extinction that threatens to take us down with it. Ecosystems, species, and life-sustaining processes are being pushed to the brink of total collapse. Plants, animals, fungi, and untold other microorganisms are going extinct 10 to 100 times the natural rate. We've all heard the numbers. 30% of birds are gone. Amphibians are on the way out. Corals are dying. 70% of insects are gone in many places. The Arctic is on fire and the Amazon is drying up. And pandemic diseases are increasing. So this is all the bad news first. Let's see. I believe that violence against nature is the gateway to all other forms of violence and injustice. It all starts with what you see here on the screen. It began when a class of people took over control of the land and hoarded all the resources, which they then used for power and control over the rest of the population. It continues to this day as the forces of capital are unleashed upon the world in the service of greed and wanton consumerism. These forces are fundamentally undemocratic. Majorities of people all over the world want peace and a healthy planet. But every time the masses rise up to demand justice for themselves and for nature, they are repressed, and regimes are installed to restore power and continue the plundering of the planet. The scene here that you see on the screen could never have occurred in a true democracy where everyone had a say in how things worked. This abomination, this ruination of the planet is made possible by scientists, by engineers, by architects, captured by the forces of greed. And we are told that emotions have no place in science, and that externalities, such as the consequences of burning fossil fuel, should never influence scientific inquiry. But a good scientist always considers the null hypothesis. What is the alternative explanation for a particular observation? What are the alternative outcomes? And a moral scientist always asks, what if this succeeds? What are the logical conclusions? In today's version of science, these are dispensed with as quaint nuisances standing in the way of progress. But what kind of world would we be living in today if science was motivated by emotion and external consequences were factored into science? Do you think the world would look like it does on the screen now? I don't. I'll give you a second to read the statement on the screen. Okay, the author of the statement on the democratization of science wants to incorporate the principles of accessibility, transparency, and accountability, equity, justice, all these catchwords that are very popular today, but doesn't actually get to incorporating the people that want those things, just talking about the concepts of those things. I would go much further in the democratization of science. I think democratization of science means reintegrating scientists into their communities where they live to solve real problems of concern to those communities, not just those who can afford to pay the scientist's salary. There is no time to waste. We all need to work together to save nature, to save ourselves. Unfortunately, we are realizing that to conquer nature is to commit ecological suicide. It's called ecocide. And crucially, this realization comes at the nadir of our connection to nature on the individual level and collectively, when fewer and fewer people recognize or know hardly anything about the plants, the animals, and the ecological processes of the planet that we live on. It has become so pronounced that some PhD students at even agricultural universities can't even recognize the actual organism that they're studying in the laboratory. So they may know all about the genetics of this organism or the metabolism, but they can't go outside and pick it out of, a field of dandelions. And like everything, this has been reduced to a catchphrase. It's called plant blindness or plant awareness deficit disorder. I think a better way to think of it is obsession with lethal technology disorder. So we're obsessed with, petroleum products. We're obsessed with all kinds of industry and applications that don't actually support life. They're not about life and they don't actually support life. And so I think we need to get back to the basics of biology, the basics of life on Earth, and those processes and activities that support life. So at the New York Botanical Garden, we've been conducting research on plants and fungi for over 130 years, amassing the second largest collection of preserved botanical specimens in the world and educating generations of scientists and promoting plant conservation around the world. And building on these institutional strengths of the garden's library, herbarium, and scientific staff, the New York City Ecoflora is leveraging these technologies like mobile computing, social media, photography, and GIS to build the most comprehensive assessment of the native and non-native wild plants of New York City ever compiled. We are putting big data in the hands of the people at a pace needed to keep up with the Anthropocene. So Charles Darwin was driven by a sense of adventure and a great curiosity about what was in his front yard, his backyard, and what was halfway around the world. And these same motivations inspired the founders of the High Line to explore an abandoned railway in the meatpacking district of Lower Manhattan. And nearly everyone today who visits the High Line, imagine many of you have probably been there. It's one of the most popular parks in the entire world, one of the most popular places, tourist attractions in New York City. Nearly everyone who visits there says, wow, I wish I could see what this was like before it became a park. Well, if you look on the screen right now, you'll see that New York City is full of High Lines. This is exactly what the High Line looked like before it became the High Line, before there were high-rise buildings on all sides and the place was totally manicured. It was wild and weedy like this scene you see here. So these are Ecoflora volunteers and they're looking for the Tree of Heaven plant. It's turns out to be a host for an invasive insect that came over from Asia 2013 or something, and is making its way across the eastern United States. And its favorite plant is the Tree of Heaven plant. And we're here looking for these Tree of Heaven. And while we were here, we found monarch butterflies. We found a rare plant for New York State, a daughter species just growing wild in this area. So it's field trips like this and other Ecoflora events that bring people together to explore the city and its amazing diversity hidden in every neighborhood. These trips and these activities also provide an opportunity for us to learn from each other and share stories and strengthen our community bonds around our mutual love for nature. Working with professional researchers, community scientists, and working with professional researchers, community scientists have made important natural history discoveries. Susan Dickerson, a resident of Colorado Springs, trained a backyard webcam on a visiting skunk that was coming to her backyard every night and discovered the animal using a rock to break the ice in her dog dish so that it could drink water in the wintertime when all the other ponds and all the sources of liquid water were frozen. This skunk knew that this dog dish always had water in it, but he had to get through the ice somehow. So the skunk picked up a rock and broke the ice in the dog dish so that it could drink the water. She caught this on her webcam. She shared her findings with local researchers, and together they published a paper on demonstrating the first tool use ever demonstrated in skunks. So a very important natural history discovery made by a community scientist with a webcam in their backyard. Eve Levine, a resident of the Lower East Side of Manhattan, discovered a wild spurge plant near her mother's apartment on the Upper West Side and posted her observation to iNaturalist, a free, non-commercial biodiversity program, which you'll learn about in a moment. Her plant was identified by Nathan Taylor, an expert in that particular plant group, who happened to be 3,000 miles away in the nowhere in West Texas. And when I saw Eve's observation on iNaturalist and Nathan's identification, I realized that the plant had never been documented from New York City before, actually never documented from New York State. And together, the three of us published a paper documenting its presence for New York State. You can see a first report of this plant in New York State. So in this way, we've discovered over a dozen new plants for New York State, citizen scientists, community scientists working together with the botanists and other scientists to make these discoveries. We could not do this alone. And these reports of the first records will be used as the baseline to study these species in the future. So one day this plant maybe will die out. It'll just sort of disappear, and this will be kind of a historical curiosity. Oh, this plant showed up and it was here for a while and then it died out. It could become the next Japanese knotweed and take over all of New York City, and this is all you hear about on CBS News. They'll be talking about this particular species. We don't know, but this is the baseline. This is the starting point. When this plant first showed up in New York City, we were there to document it. We saw that. So far, New Yorkers have made just about 300,000 observations of plants just in the 5 boroughs of New York City. You can see those don't look like 300,000 dots, but they're dots on top of dots, right? There's a lot of dots on this map. And the #1 plant observed in New York City is the white snakeroot plant, which makes me very happy because it's an indigenous species to New York City. And it has a lot of insect relationships. It supports a lot of insects in our area. And there are 17,800 observations of the white snakeroot plant. That's half of all the observations of this plant made in the entire North America, just here in the 5 boroughs of New York City. So we've been really busy and we've been really keeping a good eye on the white snakeroot plant. We know a lot about this plant, which is great. And since 1800, there were only 25 preserved plant specimens of white snakeroot made over the course of 220 years. So if we were looking at a map of preserved botanical specimens, we would see 25 dots on this map instead of 17,800 dots. So one of our goals is to get closer and closer to the true distribution of the plants in New York City. You can't just, throw darts at the map or ask somebody. There's nobody who knows where all these plants grow. It takes time to build up this data set. And that's where you all really come in strong, is you can help us cover this map with dots all over the city. And just bring to your attention the open circles. You can see them over on the left-hand side, kind of over in Weehawken, New Jersey. You see them going down into the water. Those are not aquatic marine plants that are observed out in the ocean. Those are rare plants that have been observed by people, but they've obscured the location so that it just comes out in a random place within a 25 square kilometer area. So it's just putting these dots randomly around on the map to protect the location of these rare plants. So you can't just click on the map and get all the data for these rare plants that you want. And this is a good point to remind people about the conference that we're having on November the 6th on the rare plants of New York State, and New York City. So the foremost practitioners in rare plant conservation in New York State and City will be talking about rare plant conservation. So if you have time, Friday, November the 6th from 10 to 12, hope you can make it. And later this year, or perhaps early next year, the New York City Ecoflora, all of us together, will celebrate the 1/2 millionth observation of plants and animals in New York City, the 500,000th observations. And it's only taken like 4 years to get the 500,000, not quite 100,000 per year. We might get there. So maybe some of you might be, if you get lucky, you'll be the 500,000th observation. It's going to be a mushroom, it's going to be a bird, or maybe a pigeon or a rat or a white snakeroot plant. And you'll probably, we'll all be around to make the 1 millionth observation of plants and animals in New York City. That'll be a real milestone to celebrate. And for those who want to take their enthusiasm and curiosity to another level, the New York Botanical Gardens Adult Education Department offers courses throughout the year with in-depth instruction on the birds, insects, mammals, and ecological communities throughout the city. We are training a corps of super urban naturalists who will monitor the biodiversity in their communities and help their neighbors engage with nature. And we have one of our instructors, Ken Chaya, who teaches birds and insects on the call tonight. Every generation must take responsibility for sustaining a healthy planet for all Earth's inhabitants. We must not leave the next generation to clean up our messes. And I have to apologize for my generation and the ones before for leaving our planet in such dire condition to the next generation. And that's part of my motivation, is to hopefully leave it not quite so dire. The problems of my generation and those before created, the burdens are too great for activists to bear alone. Assaults on nature occur locally, or at least originate locally. And it's at the local level that activists have the most power to affect change. You think about pollution going into our waters, pollution going into the air, organisms being killed off. All that is happening at the local level. Somebody lives near that waterway or that pipe or that road going through. We all have opportunity to engage and affect these changes. And we created the Neighborhood Naturalist Program, pioneered by the New York City Ecoflora, that meet regularly to discuss local biodiversity concerns. And because nature has no seat at the table, neighborhood naturalists act as the voice for nature in their communities. They also help educate their neighbors and inform policymakers who rarely consider the needs of plants and animals apart from human needs. And recognizing that most environmental degradations happens at the local level, neighborhood naturalists attend local board and council meetings and speak out when nature is threatened and when there are opportunities for positive change. To date, the Upper West Side Neighborhood Naturalist Group has promoted the use of bird-safe glass in new construction, the use of native plants in landscaping, and recognition of light pollution as harmful to insects, trees, and other organisms, including ourselves. Their neighborhood naturalists are a regular presence at board meetings, and they faithfully advocate for nature at every opportunity. This is something we can all do. The New York City Ecoflora is the first of its kind real-time flora built by scientists and sustained by community participation. With our botanical garden partners across the country, represented by the logos you see here, Chicago, Denver, Phoenix, and Sarasota, we are building the scientific literacy and community engagement needed to protect our wetlands, our forests, and our urban spaces that support life. It is a global model for documenting biodiversity and ecological dynamics to build a sustainable future for the well-being and inspiration of all. And we are grateful to the Institute of Museum and Library Services for their generous leadership grant to the New York Botanical Garden to support the Ecoflora. And we're almost, oh, okay, we're not doing too bad on time, right, Regina? Okay. I like to say there's no bad plant, only people doing bad things, like degrading the natural environments and introducing invasive species. It's not the plant's fault. We're the ones who degraded the environment. We brought them over here and turned them loose. And since we made the mess, we have the obligation to clean it up. And every New Yorker can help the environment by learning more about the plants and animals we share the city with, and by helping document their distribution and diversity. Each month, we sponsor an EcoQuest challenge focusing on a particular plant and its role in the environment. The October EcoQuest challenge is on mugwort. I know every one of you have seen this plant. You might not know the name of it, but might not recognize it. I know every one of you have seen it. The October is a species, the mugwort is a species from Europe and Asia that can take over highly disturbed areas and form a continuous monoculture. Natural ecosystems are inherently diverse and often dynamic, and with some species thriving for a time and then other species taking their place. That's the dynamic, then fading as conditions change. But monocultures, on the other hand, are like the meadowlands of New Jersey, where you see that just sea of, it's all one species, the common reed, or fields of mugwort, this plant you see on the screen here. These are extremely rare in nature. You only find them in the most extreme habitats where only one plant can survive this particular habitat. But in these extreme environments created by humans, and when you introduce an invasive species that's escaped from its natural predators, its fungi, bacteria, the herbivores, the animals that eat it, it's able to proliferate and totally take over the environment because it's adapted to this particular conditions. There's nothing to keep the population in check, and they drive out all the other diversity, totally eliminating local biodiversity. And every one of your observations in New York City will be used to map these species throughout New York City and help inform efforts to restore healthy ecosystems. And I thank you in advance for your service to the entire New York City community, and especially those that are neglected and most vulnerable, including the human and the non-human populations. And now I'm going to show a very brief, oh, one more thing. The woman here in the mask is Susan Hewitt, and she's the #1 observer of plants and animals in New York City. As of about an hour ago, Susan had 55,563 observations of plants and animals in New York City, and 2,088 species. That's way more species than anybody else in the entire city of New York has observed. There's nobody even close to the number of species found in New York City as Susan's. And Susan doesn't miss anything. She's got eagle eyes. And in this photo on the left, you can see Susan kneeling next to a population of tropic croton that she discovered at the edge. You can see at the edge of this basketball court at 106th Street and 3rd Avenue. So if you are going to look for the most unusual plant in all of New York City, I don't think you'd go to 106th Street and 3rd Avenue to a basketball court looking for rare and unusual plants. Susan does. She's everywhere, and nothing misses her gaze and her knowledge of things. She can see it from a block away and know that that's something interesting, and she'll go and check it out. Her observation of this plant is only the second sighting in all of New York State, and confirms that the plant is completely happy and established here in New York State. So we're right now writing this research up to document this and have this as another publication of another new plant discovered in New York State that's made its way. This plant is actually indigenous to the Gulf Coast of the United States. So it didn't come all the way, across the Pacific Ocean. It just made its way up the eastern seaboard little by little, and made its way finally to New York City. It's probably going to be up in Boston before too long. So look at the screen, the picture on the right. Can you all see what she's pointing at? You can unmute yourselves if you want to call it out, or you can put it in the chat. Justin saw it. Regina, do you see it? What Susan is pointing at? So Susan doesn't miss anything, including the opportunity for humor and just interesting cultural, everything catches her eye. She didn't miss this sign here. Yeah, so all of you probably see it by now. Notice the spelling of the sign there, restaurant. She found that. And right below her is another new plant for New York City. We don't have time to talk about all of them. So Susan is amazing. We're so lucky to have her. She's one of the top observers in the world, top identifiers. And we're really lucky to have her in our community right here in New York City, helping us document the flora. So I'm going to end here with just a little short video, and then I'll be glad to take your questions. So I'll start the video now.
Okay, so you've installed the iNaturalist app and created an account. Time to get outside and record your first observation. Here's how to do it. Any living thing, like a plant, animal, or fungus, can be an observation on iNaturalist. Once you find something you'd like to record, just tap Observe and take a photo. You can review your picture, then hit Next if it looks good. To identify it, hit What did you see? If you have an internet connection, iNaturalist will suggest 10 visually similar species and often a common ancestor. You can choose one of those or search for a species name. On this observation detail screen, you can add more photos of the same organism or write a note. The date, time, and location have been automatically added. You can also change the geoprivacy of the observation, mark whether it's captive or cultivated, or add it to a project. Once you're finished, just hit share and your observation will be uploaded for everyone to see and identify. That's it. Keep on exploring and sharing.
Okay.
Okay, so thank you, Daniel. That was
Great.
awesome. That was so great. So let's open it up to questions. I see we have one question already. And so if you have something, just put it into the chat. If you feel like you can interrupt us and ask it, go right ahead. So Madeline Mignon, who is one of the professors at Dominican, she asks, is this study limited to New York City? Or are there other studies for Westchester? And I'm sure my students are wondering about Rockland County also.
Yeah, we developed, as I mentioned at the beginning, we kind of started with Central Park and got Regina's help on that. And we next took it to the next level of New York City, 5 boroughs. And now with the help of support of the Institute of Museum and Library Services, we are prototyping or developing the model at 5 other botanical gardens in the United States. And it's a 3-year project, and we're now just starting the 2nd year. And so these other botanical gardens are using our prototype. They're trying things out, they're experimenting, they're innovating our ideas and adapting them to their local conditions. And by the 3rd year, our goal is to have a toolkit, is a kind of a compact, easy to follow standard protocol that any institution, any community, any entity can take this off the shelf and develop it for their own community. So, it might seem like there's a lot of moving parts and a lot of, you need an expert at this and you need experts to do that. We want to simplify all that, get it down to the basic elements that it can be portable and adopted in any community of any size, large and small. So you can follow along in New York City. Regina's already doing a biodiversity study of the campus of Dominican College. I think you've got students working in Rockland County. So you can already start collecting the information and working on the same sort of thing in your own community. You don't have to wait for us to get started. The nature's out there waiting for you.
Yeah, I made 2 projects. One of the things in iNaturalist that's nice is that you can make projects. Mm-hmm. And so like Daniel said, I did one for the campus, but I also did one for Rockland County. And so you can follow my project and see everything that's been documented in Rockland County. So yeah, like Daniel said, feel free to do that in your neighborhood.
Can I add something? I'm sorry to interrupt.
No, go ahead.
Lower Hudson Prism also has, I forget what they call it, but they have an EcoQuest kind of project every month. I'm sure Daniel knows about that. A guy named Brent Boscarino runs it. And sometimes they are using the same plant and sometimes not.
Right. The Lower Hudson Prism is focused on invasive species. And every month they have a focus on a different invasive in the Lower Hudson region, which includes all of Westchester County and some of, I guess, all of Rockland, some Orange and Putnam County. So most of our quote, listening area that we're in right now is included in the Lower Hudson Prism. And if you're at all interested in invasive species, they're the go-to organization for that.
Well, that leads nicely into the next question from Regina Ryan. She says, is there anyone studying the vines shrouding and killing trees along the parkways leading out of New York City to Westchester and beyond, the Hudson and the Henry Hudson and the Taconic? And as far as I know, a lot of those are porcelainberry vine and others.
Right. Unfortunately, there's two that kind of work together, the porcelainberry and the bittersweet. So the bittersweet goes up the middle, twines around the stem, and the porcelainberry goes up the outside, and together they just totally destroy a forest. And that's been a vexing issue for a long time. That, you know, I think part of the problem is that there's a jurisdictional issue. So everybody, you know, says, well, that's not my property. That's not my area. You know, oh, that's Metro North, or that's the DOT, or, you know, Parkland, or it's not my responsibility. So I think that's one of the biggest obstacles to dealing with invasive species in our area. The state of Washington has a great legislation where you're required to remove these invasives from your property. So I encourage you to look into that, the state of Washington invasive species regulations, and advocate for that here in New York State. We've got a long way to go before we get to that stage. But one of the things that we're trying to do with the ecoflora is prevent the next Japanese knotweed, the next porcelainberry, the next bittersweet from ever getting to the stage where it's like so overwhelming that nobody knows what to do. I mean, what can you do? I mean, you just spray the herbicide out of a helicopter and just kill everything, or, you know, there's hardly enough resources to get rid of so much of that. So one of the objectives we have is to stay on top of these things from the beginning, to nip them in the bud, pun intended there, before they get out of hand like the porcelainberry.
Doria Hillsman asks, in the first slide, you stated that we are at the peak. How do you know you've reached the peak until you see a decline? I didn't see any downturn at the end of that line.
Yeah, in that graph.
Yep.
Yeah, well, that ended at the middle of the Holocene 10,000 years ago. So the last 200 years is not on that graph.
And that's where you would see the decline.
Right. Right. That's a good point. I probably should start putting that on there. Would be very instructive. Depressing.
Sandra Powers asks, is there something published about light pollution and how to make it less damaging to plants, a type of light maybe? Also, are there types of plants that are more sensitive or affected by light pollution?
I know there's been studies of trees and their impacts. They've done research that show that trees actually sleep. They think that we can detect this because the branches sag down a little bit, and they have their metabolism slows down at nighttime when it's dark. And so, you know, that kind of looks like sleep, and probably is something similar to it. And light pollution disrupts that circadian, that natural rhythm that goes through the day. So it's been studied in trees, the effects of nighttime light pollution. And as far as what type of lights, I know that these blue lights, the LED lights that are used more and more frequently now, are especially damaging to, they go into the retina, and they're much more powerful disruptor of our circadian rhythms than even those orange sulfur lights. As difficult as they are to look at, they're less damaging. So these really bright, more blue lights are, I've read that they're more disruptive to organisms. So those are two examples. And there is quite a bit of literature. So you just, do some research and you'll find papers published on the effects of light pollution on organisms, plants and animals.
Daniel, does somebody have a question?
I do there. Hello. And
Hi, Jill.
hi, Daniel. I just wondered if you could share with us what the responses have been so far to the EcoFlora project in terms of other organizations in the city, such as the Parks Department, or if you've discovered people that are utilizing the information to positive ends.
Yeah, great question. I'm glad you asked that just now. When we were doing the study of the plants of Central Park, over 3 years, we covered every inch of the park, looking at every plant. We found lots of really interesting things that had never been seen there before, things that had thought were lost, rediscovery of things, lots of stuff. We were all learning very much. The three authors are right here, Regina, myself, three of the authors, and Ken Chea. And one of the plants we found, we just, one of the places that we had not gotten into to look at in Central Park, and we were trying to cover every inch of the park because we knew, people were going to ask us, oh, did you go there? And, did you get at the baseball fields? And did you go on those islands in the lake and all these places? One place we had not been was behind the Delacorte Theater, where they do Shakespeare in the park. And it's it's all fenced off. You need special permission to get in there. And we kind of were putting that off. And eventually we got permission. We decided, today's the day we're going to go over there and see what's growing underneath those bald cypress trees behind, Hamlet when he throws the knife into the water or something. What plants are there in the background of that theater. And we found this very tall grass that was taller than me. And I recognized it from my youth growing up down south. And it turns out that it's a highly invasive species that had only been seen in New York City a couple of times. And if this plant got loose in New York City, would be a total disaster. Would be like mugwort and Japanese knotweed combined together to just take over. And it will grow everywhere, salt marshes, in lawns, and all kinds of habitats, and crowd out native species. So we informed the Central Park Conservancy right away and let them know that this plant was there, and they came along and got rid of it. So, that was using this information for early detection and rapid response to the infestation of an invasive species to protect all the rest of the biodiversity in New York City. So unfortunately, I found the plant again up in Morningside Park, and with a group of interns up there, we had a great morning, pulling it all out and learning about Johnson grass. But it takes vigilance. We need to stay on top of it, and we all need to learn to recognize these things. And with your participation in the EcoFlora, with learning about the plants and the animals in your communities, you'll be able to spot these, and you'll be able to bring this to the attention and help protect our biodiversity.
Daniel, can you tell people how to join the EcoFlora project?
Yeah, you go on iNaturalist and you search for New York City EcoFlora and join the project there. And start making observations. Just go outside with your phone and Take a picture of any living thing, upload it, and see what happens. You'll be amazed at the response you get, I'm sure.
It's really fun. It's very addicting.
Yeah. And family friendly, very safe. Totally free, non-commercial. They don't sell your information to anybody. It's open source. You can download everything. You can set your privacy settings. So it's very safe, non-commercial, and no danger at all. Great. So is that it for the questions?
Any other questions?
I got a question.
Go ahead, Jaden.
Since you said you've done some like findings up in like Putnam County and everything, have you ever thought about doing any like findings for like any mammals, anything out in like Sullivan County? Because like I'm kind of from like upstate New York, so have you ever thought about doing any like observations out there?
Well, I was actually in Sullivan County not too long ago looking at ash trees, looking for resistant trees that are resistant to the emerald ash borer. So there is a lot of biological research happening in Sullivan County right now. There's lots of scientists working on the water quality, working in the Adirondack or Catskills Park to monitor the biodiversity. And Jaden, you could help them by observing the plants around you in your neighborhood. And this will inform everybody who's working up there in Sullivan County to protect the biodiversity there. So you can immediately become a participant in the protection of the biodiversity there. So don't wait for me to come up to you. You can get started right away.
And Jaden, I can show you on your iNaturalist how to find the observations already made in Sullivan County.
Mm-hmm.
Okay.
Yep, thousands of them already made.
Anyone else have a question?
Okay, well, thank you so much for listening. You guys were a great audience. I really can't wait to meet you all in person. Maybe the next crop of students, but hope to see you online and hope to see you out in the park observing nature.
And I put in the chat, and I will send it in the email, the information about the Ecoflora conference,
November the 6th, 10.
November 6th, 10 to 12. So
That's also, is that also going to be virtual?
Yes, it is.
Yes.
Yes, online.
I really enjoyed this. Thank you so much.
You're welcome.
thank you, everybody. Were you getting lots of thank yous on the chat? So
Great, thank you.
I'll stay online if anybody has any other questions. If not, thank you, everybody, for coming.
Thank you so much. Have a good night.
Thank you. You also. Thank you, Daniel. That was great.
You're welcome.
Wonderful.
Your students are so great.
Oh,
They're
it's so wonderful. And a bunch of them were really here. It was really good.
yeah.
And a bunch of my students are doing iNaturalist, so this I hope inspired them to do some more, and like Jaden to do his own area, not just on campus.
Yeah, come down and meet Susan Hewitt.
No,
Our-
that's amazing how many observations she's done.
yeah, and so many of them are so interesting. She's got a great eye.
I can't believe Croton is growing in the city.
It's actually a new genus for New York State.
That's
No other species
amazing.
of croton, first one ever. Yeah,
That's incredible. Thanks for coming, Kevin.
yep. Good to see everybody. Good to see your names.
Yeah. Wonderful.
Yeah.
I do have a question. I know both of you have been working on Central Park for a long time, and I wondered if you're pleased or surprised at how the native plant ideas that were put into practice many years ago, seems that there has been something wonderful that happened in terms of the Ramble and the other areas in Central Park around the water bodies and in the woods. And I think it's perhaps one of the, to me, it seems that it's a tremendous success. So I'm just wondering, it's kind of pioneering, such what you did. So I'm just wondering what your feelings are about it.
Well, I'm just going to jump in before Regina does.
Go for it.
Give a shout out because Regina was the head gardener in Central Park for 19, not all of those 19 years, but she and her colleagues really started, Neil Calvanese started the emphasis on native plants and restoring these areas that were overrun with invasives. So managing the invasives and encouraging the native plants to come back. It took a long time, but I think our data shows that it was worth it. It paid off. There was a 5% increase in the native plant diversity in Central Park over the 10-year period that we know of.
Yeah.
And
Yeah, it was really wonderful to see and to, because like working there, that was my passion, was to encourage the native plants, to put in native plants when we were planting, get rid of the invasives, but to see it like in a study, one study done in 2007, and then the next one that Daniel and I did, and Ken and everyone in 2017, and to see a 5% increase in the wild native plants, not the stuff that we planted. I mean, the stuff that we planted started regenerating and became wild. But Daniel and I, that we were looking at the wild plants, so it was very exciting to see that and to get that confirmation.
So you mean additional plants to the ones you had planted appeared there, or they just?
So some of the plants that we saw, and Daniel can chime in on this, you can't always tell how they came in, but plants are mobile. The seeds come in on the wind, the seeds come in on the feet of birds,
Yes.
they come in on our feet, they come in a truck, whatever the means is,
Oh,
the seeds come in.
yes.
And so we found some really interesting native plants that I know we didn't plant,
Wow.
but that were there. So either they were always there or they came in somehow.
Wow. Oh, yes.
Would you agree with that, Daniel?
absolutely, like the pumpkin ash.
The pumpkin ash. Yeah.
Yeah, nobody planted that.
Mm-hmm.
Hmm.
Maybe a beaver 10,000 years ago or 300 years ago. They all got killed off
Thank you so
by
much.
John Jacob Astor. Beaver killer. Okay, shall we wrap it up?
Yeah, I think we can wrap it up. Thank you, everybody, for coming. And thank you again, Daniel, and for staying a little extra to answer some questions.
Sure, yeah.
All right, so see everybody. I'm going to stop recording. Bye, everybody.
You can see I'm in my office. I'm going home.
Go home, Daniel.
It's time. Good night. Bye.